Alright, so let's get us started with just a couple slides to finish up our unit on DNA and how we get that information. And it's stored right here. You can look at this molecule up at the top in a double helix. How can we get that information out of the nucleus in a form that still is meaningful and can be used, and to make one last step called protein? And so that's what we've kind of been learning about in class. Our topic today is, well, what happens if one of these steps goes wrong? Somewhere in the process, the enzymes that we learned about that are the spell checkers, what happens if they don't do their job? And so I just want to show you a little bit of an example here, how one difference in an amino acid can change an entire shape of a red blood cell. And it's the 6th amino acid right here that we're looking at that's highlighted in pink. If that's the amino acid that changes, what happens to our blood cells? So it's pretty interesting stuff that we're going to look at. Alright, so there are a couple different types of mutations just based on what gets switched up. So the first one we're going to look at here is something that's called a point mutation. And what that means, what's happening, is at a single point or a single location, you have a single base change that occurs. So I've got a little animation up here. Wild type, we'll learn this with our fruit flies when we get a little more into heredity. Wild type normally stands for something that just means kind of true breeding or even normal. And it's oftentimes represented with a plus. So just think of that, something to store for later on. So here we've got a very short protein. It just has 4 amino acids, and then we learned about our stop codon. So this is the strand of messenger RNA that we're looking at. So what would happen if we would change just a single point? And so I've got a single point that has changed right here with a uracil instead of a cytosine. And so at that single point, there's 3 things that could happen to this protein that we make. If there's no effect on the amino acid sequence, if you get the exact same amino acids that you got before, we call that a silent mutation. And probably the majority of the mutations that are happening in your body are silent mutations. Evolutionary-wise, there is a little bit of redundancy in our code. There's multiple ways of getting the same amino acid. So this is kind of built in to our genetics in a sense that we can tolerate some mutations. Now, the other thing that can happen is if we change an amino acid, we call that a missense. So if we look up at the example here, we have an A that's been put in instead of a G. And if that occurs, we have AGC, and that changes your amino acid, and that's called a missense. The last one, if you would have a change that would occur that causes a stop codon, like this one and the third one here that I'm showing, that's called a nonsense. So let's kind of start hinting about some of our evolutionary ideas that we're going to get into in our next unit. When do mutations affect the next generation? So think about that today, and we'll probably talk a little more about that in class. Alright, so let me show you a little bit about a point mutation, and we'll go back to that reference that I showed you at the beginning of class with sickle cell anemia. So here is our wild type hemoglobin, and here is our mutant type hemoglobin. So we can see that mutations don't have to just happen on messenger RNA, they can also happen clear up here on DNA too. But because DNA, what we've learned, makes our messenger RNA, that A now becomes a U when it should have become, when you should have had a T on the DNA, which would have become an A. So that changes an amino acid. And if you answered that it was a missense mutation, you were correct. So one little boo-boo can take transcription and translation and change your protein product. Alright, now point mutations are kind of the easiest ones to look at, and probably the ones that cause the least amount of changes. Frame shift are much more dramatic because what happens with these is it's a shift in the reading frame. Now we've learned, and we'll look up here, we pretty much read these in groups of 3. We called them codons, and we said it's in a triplicate code. What a frame shift is going to do is it's either going to add a base or it's going to delete a base. So we've got a deletion up here and we've got an addition of a base down here. And what that does is the point where you either have something added or deleted shifts your reading frame. And it will impact from that point downstream. And the same thing happens here. Just like we had nonsense before that caused a stop codon to be put in, that same thing can happen. You can actually have an entire triplicate inserted or deleted also. It doesn't just have to be 1. You could insert 2. You could insert up to 3. But as long as it's an insertion, that means we're adding bases. And if it's a deletion, that means we're losing bases. And what I meant about this downstream idea is it changes everything from that point on. The whole reading frame is different. So here's a question for you. Where would this mutation cause the most change? If it would happen at the beginning or the end of a gene? So if we look at another interesting case with some genetic disease, again, cystic fibrosis. A gene for phenylalanine 508 is completely deleted in patients that have this, and what this causes is instead of having a protein that can help move mucus along through pathways and ducts, mucus actually builds up. And so that's what you're seeing. These parents, these little youngsters have cystic fibrosis, and they actually have to kind of hit and pounce on the chest to break up some of the mucus so that the kids can breathe easier. So just one little change on chromosome number 7 can really lead to some things that are pretty serious. So that's pretty much it with mutations. They're pretty short and sweet, pretty easy to get to. We'll talk a little bit more about mutations with evolution. So I do want to talk a little bit about prokaryotes, just to get us an intro into where we're going next with some of our DNA technology. We've always talked about how we get the DNA information out of the nucleus. So what happens if you don't have a nucleus? Where does transcription and translation take place? Well, you got it. Right inside the cell membrane. The only thing that you've got holding in here is cytoplasm, and that's exactly where this is going to happen at. So let's just look at a little difference between prokaryote and eukaryotic genes. Prokaryotes, DNA is in the cytoplasm that I just mentioned. Eukaryotes, it's in a nucleus. Prokaryotic DNA is circular, looks like this. Eukaryotic DNA is very linear, double-stranded like this. This is also double-stranded. I don't want you to think that it's not. Ooh, this is a pretty poor double strand. But it is circular in nature. This can be coiled up into a chromosome. This does not get coiled up anymore. When we coil it up into the chromosomes, we utilize something called histones. It just helps to keep everything organized. Prokaryotic DNA does not have any histones. We say that it is naked. We also talked about how we do some processing of our message. Eukaryotes have introns and extrons. Prokaryotes, no introns. So much more simpler DNA system. And so that makes translation a lot more simpler too. This happens at the same exact time. It's simultaneous in bacteria because it's happening in the cytoplasm. You don't have any messenger RNA editing, and you have ribosomes that read the messenger RNA as it's being transcribed. So that's what you're seeing here. These are all different types of ribosomes. Here's your RNA polymerase. And from that point on, as soon as you make that messenger RNA, you have tons and tons of ribosomes that read it. And we're just moving down along the DNA strand. And that's what this picture is showing. So if we sum up what our differences are, it takes a lot of time for eukaryotes to do their processing, about an hour to go from DNA to protein in a eukaryote. I think you would find it interesting that if E. coli are going to completely divide in 20 minutes, then they must be doing this a lot faster than eukaryotes. Also, we have a barrier. We've got the nucleus and the nuclear envelope that we have to contend with in a eukaryotic cell. Prokaryotes, you don't have that, so it's a much faster process. Alright, so just wanted to wrap up those few things, and what we got to start thinking about next is what's the value of having a mutation? What's the value in making a protein that's brand new to a system, and how does that impact the organism and the cell? Alright, as always, make sure you bring all your questions to class, and I will see you soon.